Good evening, students. Very warm welcome to the ninth day of our workshop. Today, we will be dealing with the bioinformatics part, a little bit about its introduction, and X-ray crystallography. These two techniques we will be dealing today. Hello, everyone. So before we start, let's have a little bit of interaction, some conversations. Tell me about yourself. How was your day? What you did today? Everything. For me, it was a busy day. I went to the bank for some work. I'm studying some courses, so I'm self-learning a little bit. Awesome. And yeah, thinking about future. B pharmacy. You can open your own chemist shop, I guess, or is it after D pharmacy? That's good. I mean, having a chemist shop is big deal. I face in the busiest shop, especially in this COVID days. Um it's one of the highest money maker shops from morning till night, Monday to Sunday. You have to work hard for that, of course, but you get paid off afterwards. You can also go for pharmacovigilance uh, in clinical research pharmacy, and so some courses like that are possible. Oh yes, drug discovery research is also a good option. So all these are possible things. Oh, MSc in biochemistry. So I was looking for some students, some free courses. So I found out post molecular biology and biochemistry, but the date has been gone for the same. But there is a very nice courses. I like the how the course schedule, the modules were. They were quite apt, and there will be a lot of learning. Though University of Potsdam doesn't have a big place, but all in all, I like the structure of the module. So afterwards, you will have for sure some future later on. And I always say check at the dart.com, because I know much more about Germany rather than India at the moment. So I can find out, I can find the way that you can look for the things that is apt for the future. Certainly. I will today not take you much of your time, so it will be a short lectures, and yeah, so I mean, after MSc in biochemistry, you can look for PhD or look for industry job, but most of the times we tend to focus on PhD because It's a 3 years project. You in the Germany, it will be 3 year project. You will be earning handsomely per month for 3 years. You can go or walk around Europe, which is the best part, and you can save a lot of money. And indeed, 3 years is a journey where you learn a lot from the experience of European life. And yeah, genetic counseling. What is genetic counseling actually? So it's good to interact with the students. I get to know about new stuff. It's given by WHO. Okay, to find out if somebody has a problem in the genetic history and provide them with the help of genetic testing and related procedures to know about it. Um, it seems if you are Darpak, if you are like good as a counselor, you love to teach students. You love to like not teach, but to counsel people and to solve their problems. Then indeed, it's a good profession, no doubt in that. But it's just it should be inner calling. What is your inner call? Is what you inner what is your inner thing is. Just don't go for money, because money you can earn. Like for me, uh, I always learn for knowledge. I always run for the knowledge and satisfaction afterwards. So teaching found ultimately found me the satisfaction. So I didn't choose the research part of my life after PhD. I choose the teaching part where I can spread the knowledge that I have gained and make me ultimately at the end of the day satisfying sleep. So I'm not running for it or making my life what do you say, difficult. I'm doing what I'm enjoying actually at the moment. So I enjoy teaching, I enjoy sharing things with students, and it makes me feel good. That should be the matter. So whatever, whether it's MS in biochemistry, whether you're doing for PhD, whether looking for gene therapy or anything, any profession, uh, you can ultimate goal should be your inner joy, your inner happiness, your inner peace. That's matters, because these things we mostly forget while choosing our career or university or subjects or something. Like we just go like bhed chal, sheep wandering. At once something started, then we all started the same way. So just keep in that mind. So which university is best for molecular biology in Europe? Which university is best for the molecular biology? So there are some programs on this website. If we put molecular biology, molecular bioengineering, International Max Planck Institute, MSc Integrated Immunology, Neuroscience, Weitran-Zeigen, Hanover Biomedical Research School, Agriculture Biosciences. So there are a lot of options coming up. And to look for, make sure you look answers on the Quora also. On Quora you get best answers for us, for Indian especially, because it's made for India. What are the best university to study life sciences or files related to biotechnology? So look for two answers. Dad, he suggested. ETH, he suggested. And it's not good one. Look for masterportal.com. Or studyportal.com. So one is Quora. Just look for your answers in the Quora. So this is the best place to go for. Quora, study portal to look for the best universities. So here you can study about which university will be best for you. So while writing a foreign admission on universities, what points? So your SOP, statement of purpose, really matters. Your grades really matters. You might be have an interview that really matters. Your TOEFL score really matters, and that's it. So these are the basic criteria. So SOP need to be strong. That why you want to study that course, how could that promote your future, your career, how it could bring benefit to university also taking you as a student. So think like that, then sell yourself. So it's all about selling your things. So I have visited many countries during PhD. So I visited Italy, Netherlands, UK, Spain for some time, and I travel a lot. France, I was there. So I had a good exposure of Europe, Poland. So all these places, Czech Republic also during New Year time. So it doesn't matter which country you choose for your genetic research. All countries are best. India is also best. The option is that how the institute is, how the value of that institute is. Just check for that. That really matters. Whether it's in India, or whether it's in Europe, or in Australia, or in Canada, or in US, or in States, anywhere. So it should be matter that it has to be uh, good quality. Good quality of work is going on there. Just check the research publications of that institute. Check how the department is, how the facilities are available in that institute, how much money they have for their work. All these matters. What is the better option to do? PhD in agriculture biotechnology in India or any other country? I mean, in India it will take you around 5 years for sure. India is also best. You get now really good paid during your PhD. Earlier it was not. So I would also recommend India is the best place to do. So I'm also quite patriotic about India. So why not we give preference to India as compared to going outside? So go for India and then look for options. Many options could be open while you're studying in PhD. You can go for exchange scholarship also. Exchange scholarships are also available to travel abroad for some months and then you can finish your PhD in India itself. You can have exchange program also, so that not to worry about. Good evening, good evening. So, on that note, I will start with the X-ray crystallography technique first. So mainly X-ray crystallography is dependent upon to know about the structure of of a protein by adding X-ray rays on your protein, and then they get diffracted on a screen, and then we calculate via computer and then make a figure out of it, computational figure, and then we are sure that our structures looks like this. That's the basic principle. But before we do that, we have to create a crystal of our protein so that the crystal could have a better image. It's a better component to have things done in a way to have better results. So in this, you will be acquainted with the basic principle of X-rays, phenomena of diffractions, how this can be used to decipher the structure of proteins. You will get to know about depth knowledge on the instrumentation and mechanistic overview of X-ray crystallography. So structure, why structure determination, how it helps us in understanding. So protein interacts with other molecules, how they perform catalysis in the case of enzymes, interaction of proteins with other molecules including protein itself, miscoding or misfolding of proteins associated with the disease. So all these are led to deal with the protein structure. And what is X-ray crystallography? It's a form of very high resolution microscopy, enable us to visualize protein structure at the atomic level to enhance our understanding of the protein functions. So it is based on the X-rays that are diffracted by the crystals. So you have a crystal here in the center here, you have X-rays and they get diffracted at some angle and they are spotted on the detector, and then we remove around this crystal and then we make a figure out of it. So then comes the microscopy, wavelength and visualization. So there are three types of uh, source that we can use: light, electron, X-rays. But why we choose X-rays, not electron or light? Because of the wavelength. First of all, it's very least 1 angstrom, and visualizations we can see with the X-rays at the atomic detail of our structures, but in the case of electron and light we can see mainly at the cellular level, so which is not more deep for protein structure analysis. X-rays in the order of atom diameter and bond length allow these to be individually resolved. No lens is available to focus X-rays. Crystals act as a magnifier of the scattering of the X-rays in this case. X-ray crystallography steps in structure determinations. So protein purifications, protein crystallizations, data collection, structure solution, structure determination. So all are these are 5 steps. So in first you have a crystal, you go through the X-ray, you have a diffraction pattern, then you make electron density map afterwards in the refinement steps, then you have an atomic model out of it at the end. So like this, you make a structure of a protein. So in step 1, protein purification, this is done with the help of SDS page. You can isolate 1 or 4 proteins from the complex mixture, usually cells, tissue or whole organisms. So why we do this protein purifications? In order to have characterization of the function, structure, interaction of the proteins. So you need 5 to 10 milligram of pure soluble proteins with 95% purity. So keep that in mind while doing so. Then comes the protein crystallization. Once the protein is extracted, it's time for the crystallization of the whole thing. So here you can see one molecule, one unit of cell, But in the crystal there will be a lot of like a box, lot of these units together, so which will help us to make a better structure. So an X-ray scattering from a single unit would be unimaginable weak. So crystal arranges a huge number of molecules in the same orientations. So scattering waves add up in the face of increasing signals to level which can be measured, and this is often the rate limiting step in the straightforward structure of the membrane. So like the X-rays will come and they will hit it there, X-rays will come, they will hit it there, X-rays will come, they will hit it there, yeah, and then after hitting they will diffract. They diffract this side, they diffract this side, they diffract this side. So based on the diffraction, we can, on the detector, this will be a detector, we can prove that the protein structure might be look like this. We can assume by the diffraction of your X-rays from this protein structure, then on the crystal afterwards, that might be your protein might be having these electron densities of protein. So crystal, in crystallization, your crystal must be less than 1 millimeter. No crack should be there, no air bubbles, nothing should be there. So that has to be taken care while doing so. So this is the first crystal grown at 4 degrees Celsius. Then this is at 17 degrees Celsius. Then this is again 70 degrees Celsius with pH 5.6. Here pH is 6. And then this is a sitting drop at 17 degrees Celsius with 31% MPH with pH 5.6. So a different pH, a different temperature, your crystal starts to form a different shapes. So this is a hanging drop method. So you put 1 to 5 microliter of protein solution suspended over a 1 ml reservoir containing precipitant solution. Example, your precipitant solution could be ammonium sulfate solution or polyethylene glycol, PEG or ACSS, AMS. Then protein are being diffracted. The reactions goes like this. This is a Bragg's law. So you might have done this in +2 somewhere in the classes. So beam of ray come and then they diffracted like this. So they make these angles, equal angles. Your BC is equal to CD is equal to D sine theta. The width of the diffraction that is happening, and it's equal to that sine theta of this. So they got the Nobel Prize winner for the same also. Then data collection, we are done with the protein purification, then X-ray crystallizations, then afterwards there comes the data collection. So this is mounting. Crystals are mounted in a way that sample can be rotated and X-ray beams can be passed through the sample. Methods of mounting including using either the capillary or tube. Both capillary tubes are mounted on a goniometer. So we expose the X-rays with the synchrotron or X-ray generators. So you have a source of X-rays via synchrotron or something, then they got reflected by the prisms, then they hit the crystals, and after hitting the crystals they are being absorbed at the area detector. So source of X-rays, synchrotron in this case. Typical size of crystals for data collections, we need 0.3 by 0.3 by 0.1 millimeter. Crystals are bombarded with X-rays which are scattered from the planes of crystal lattice, and the scattered X-rays are captured as a diffraction pattern on a detector such as films or an electronic device. Rotate crystal through 1 degree and require XRD pattern. If XRD pattern is very crowded, reduce the degree of rotations. Repeat until 30 degrees were obtained, then sometimes 180 degree dependent upon the crystal symmetry. Lower the symmetry, more data required. For high resolution, use synchrotron. So this is the data that you get afterwards, the diffracted data, typical diffraction data, protein crystals, and then a 3D structure was obtained using the electronic density map of the crystal. Now there is a 3D structure of it done afterwards. So today's outcome is that you are able to understand the topic of X-rays, applications in proteomics, principle of extracrystallography, and understand the diffraction data, how we obtain this data. These are the references. That was a short and crisp. So I will play some videos now with X-ray crystallography. So I hope what have been taught here is being clear now. How the X-ray diffraction is done, how this method is done in pure basic way. So last video is about now X-ray diffraction. So just to explain how the X-ray diffraction in the X-ray um, collection of your data works out. So, with that exciting music, Bragg's law has been explained in a good way. That's awesome. Yes. So, students, we are done with the X-ray diffraction part for today. Would you like some break or we continue? Shall we continue? Okay. So, I will take you to the next part of today's lecture, that is bioinformatics. So, name itself suggests that something to deal with informatics, that is IT skills and biology. So, computational biology. So, all the data that is related with the sequencing, algorithms, computational data, sequences, software, evolutionary complexes, phylogenetic studies. So, all these things are coming up in the bioinformatics. So, this is a tree, phylogenetic tree, kind of a tree with the words being showing you, letting all the protein and bioinformatics part coming together, and there's analysis of it. So some biological concepts that have been that's we deal here. So outline of this today's lecture would be some biological concepts, a brief history of young field, where is all data coming from, and large scale biology, new words, new techniques. So that's the basic outline. So as I explained, that bioinformatics is something to deal with the biology and biophysics and mathematics. So coming all together parts of statistics, informatics, mathematics, biology and biophysics is coming out in this Venn diagram, showing that bioinformatics belong to all these parts. So if they are studying, so bringing them all these three techniques will be the one that dealing with the bioinformatics. And then further, so we have a from the DNA we can extract DNA and genome database. From RNA we can have a complementary DNA transcriptome. Then from protein you can have protein sequences, structural databases are being extracted. And then from whole of this you can have a phenotype, or the from the protein itself you can understand about the proteome at the end. So, according to the National Center for Biotechnology Information (NCBI), and it says that bioinformatics is the field of science in which biology, computer science, and information technology merge into single discipline. The ultimate goal of the field is to enable the discovery of new biological insights, as well as to create a global perspective from which unifying principles in biology can be discerned. So there are three important subdisciplines within the bioinformatics. First is new algorithms and statistics, then analysis of interpretation of various types of data, and then third one, efficient access management of different types of information. So bringing all these three as a paradigms of the bioinformatics. Bioinformatics deals with the biology, computer science and information technologies merging into a single discipline. So tree of life of three domains, archaea, eukarya, bacteria. So all these three belong to the three domains of life, and we also study in bioinformatics and phylogenetic studies. So bacteria, archaea, eukarya, under which we had different phylas, Dipan and Locaryata. So according to them they are defined as. And in our studies of our bacterial and eukaryotic cells, there are two types now, bacterial and eukaryotic. So bacterial cells, as we are now aware of, that their structures looks like this, and eukaryotic have different compositions from the bacterial one. And central dogma of life means from DNA your RNA is made, from RNA then your protein is made. So in prokaryotes it's different, and in eukaryote it's different. In prokaryotes you have a gene A, gene B, transcription is done, coding region A, coding region B, translation is done, then protein A and then protein B are produced. Then in eukaryotes here, you have a DNA, but it has introns also. So these introns in the primary RNA transcript, they are spliced, they are moved, so that at the end you have mature mRNA. Then mature mRNA is transported to the cytoplasm, mRNA, messenger RNA, then translation is done, then protein is produced for your cytoplasm thing. So I've told you already in the protein splicing, on the left side we have extine, intine, extine. So your inteins are moved, protein splicing is done, you have mature protein. In the case of eukaryotics, you have introns, introns are moved, mRNA is spliced, you have mature mRNA at the end. So bioinformatics is about genetically coded information and proteins. So what we have discussed so far in the prokaryotes and eukaryotes, basically in bioinformatics we will be dealing with the genetic codes, like how much genetic code has been transferred from one cycle to another. So in this, like in this case, we have DNA, we have a RNA, and then we have a protein. DNA could replicate, self-replicate in this triplicate. These are the triplicate codes, and then transcription is done with these genetic codes to RNA. And then further translation is done with these amino acids: phenylalanine, valine, aspartic acid, glycine, histidine, leucine, like this. So bioinformatics is about data management, database analysis, and interpretation of data using web-based or standalone tools. So we also use Python language, Java, Perl is being used in this, and development of new algorithms and statistics. So in summary, bioinformatics help us to uncover biological information hidden in the mass of sequence, structure, literature, other biological data, obtain an insight into the fundamental biology of cells and organisms. So from genome, transcriptome, from transcriptome you can have proteome, from proteome you can extract metabolome, or metabolome you can get to know about the phenotype. So all are interconnected, so everything is very important and play a crucial role in identifying the things. So these are the computational tools and infrastructure on the y-axis and their biological complexities on the x-axis. Comparative genomics is one of the simplest one, and it doesn't have much complexity. Then comes the constraint based digit docking, so that is done in the drug discovery case. Then comes the genome scaling, protein threading, to know about genome level structures. Then comes cell pathway network stimulations, you do with the stimulation softwares. Then constraint based flexible docking. Then comes molecular machine classical stimulations with the help of softwares, like very complex softwares you can do in the computational. It's mainly for the computational biologists who are working in there. So for them it is quite important. So by this way it's getting complex and complex at the end. So data in the DNA case, raw DNA sequence information we need. 4 bases are required, A, G, C, T, whether they are coding or not, whether they're parsing into the genes, genetic codes, and the signals and patterns at the end of the studies. So like here in this case, we are concerned with this A, T, G, and this C, G, G, C, A, G, G, C. So these are the coding part that we are concerned. So this, in this case, bioinformatics tools might be helpful to identify them and to work with them, whether you want to mutate them or mutate them or transfer with another genes or remove them or add some another gene to it. So it's up to you. You can do it very easily first on the computer, and then you can start doing it at the molecular level. So that's why bioinformatics plays a big role in this case. So restriction enzymes are sequence specifics. They recognize and bind to specific DNA. And enzyme E. coli, for example, it binds to GATTC. So how often do you expect E. coli to find its recognition site? So within this sequence, if you want to find how many times E. coli will attack and make a GATTC, this palindromic sequence, so only in this time here. So this could be done with the help of software identification. So you can do this if you have a big genomic data, and you have to find out that this restriction enzymes E core I1, where it will be cutting it. So you can do it with the help of bioinformatics softwares. So something about basics. Adenine, guanine, purines are the larger of the two types of bases found in DNA that we have discussed it already in the first classes. Thymine and cysteine being pyrimidines. Base pairs within the DNA complex, A form two hydrogen bonds with T, and G forms three hydrogen bonds with the C on the opposite strand. First letter, second letter, third letter. So this is how you make your genetic code. So UUU, UCU, UCA, UCG like this, and then there is a stop codons, URR, UAG, UGA. Then there is a methionine as a start codon, AUG. And these protein sequences that we have discussed, so total in our UniProt or SwissProt, this is the database, big database of our protein data. Taxonomic distribution is done there with the protein sequences. So bacteria has around 60%, then comes archaea, then comes the viruses, then comes the eukaryota. So like this your data is being divided. I think this is eukaryota and this is archaea, sorry. Archaea being 4%, 3% viruses, 33% is your eukaryota, 60% is your bacteria or in your UniProt database. So if you go to the PDB website, protein database website, so there is every month PDB molecule of the month. You can have a look there on the website and get to know about who is the PDB molecule of the month. So this is possible by the website. So some historical landmarks: in 53 structure of DNA was revealed. Then in 66, in 60s elaboration of the genetic code. Then in 80s gene database is set up. Then in 90s the term bioinformatics appeared to the scientific publications. Then in 20s first draft of human genome was produced. Then in 2003 mainly the human genome project was completed. So mainly all the studies related to bioinformatics on the PubMed and CBI are all in the world from everywhere respect to database in bioinformatics. It started to study after the human genome project has been successfully done. That is in 2003. So bioinformatics, that is your computational biology, computational molecular biology, biomolecular informatics, biocomputing. So all mean same. So whenever all the PubMed, if you in the PubMed put bioinformatics studies, you can see the resulted year initially by the 2000, it was nothing much. Hardly 1500 to 1600 studies were done. So but in by 2003 when they have discovered about genome data, so it's rapidly start to increase rapidly. So a lot of things starts to come to deal with this part. Then in 70s algorithm for the sequence comparisons was done by Needleman and Wunsch, and further in 80s Smith-Waterman algorithm for the sequence alignment. Then 80s 8 Pearson and Lipman fast search for sequence database. In the 90s basic local alignment search tools SciBlast are done in that case. So here showing you data about the gene bank. It's again from the 2003. It started to take exponential curve and your gene bank started to increase in very high amount. So in 2017 recently we have this much of 2 raised to power 10 2 into 10 raised to power 11 base pairs and specific sequences are present. So these are the National Library of Medicines. So how base pairs were find out on the y-axis in the year, downside is the axis is the years. So base pairs in millions. So in the PubMed, so it was PubMed first. The BLAST was discovered 90s, then Entrance, then GeneBank, then Genomes, Omim, PubMed, Human Genome. So all the various data banks they started to come. So again, in from the 2003, they started to be rapidly up with these various discoveries done. So like 1000 genomes, uh, genetic testings is were done, medical genome pub reader was developed, genome reference consortium was done in 2007. These are all data over the 24 years of growth, NCBI data and user services. Then yes, DNA sequencing with the chain terminating inhibitors with the Sanger technology. In 1977, they have discovered it. That's changed the totally way of checking the DNA sequencing. So it's a high throughput sequencing technologies, that is next generation sequencing methods. So in 70s, organism bacteriophage was first sequenced. Then first living organism Haemophilus influenza HIV was sequenced in 95. In 96, first eukaryotic Saccharomyces cerevisiae Baker yeast was sequenced. So from there, all the basic changes started to happen. Then basis sequential capabilities versus computational power. So you can see that in the green line showing you the computational power, which is being going in straight forward but increasing uh, every 18 months, whereas your sequencing capabilities, they are increasing after every 4 months. So that's somehow the computational versus sequencing are correlated. Then comes the sequencing read archive database growth, where we can see that the next generation sequencing technologies, they started to produce very huge amount of data within the span of 5 years. From 2010 to 2015, the sizes of the basis pairs are increased tremendously. Then complete genome sequence available in the public domain was published as per this. So cumulative number of genomes are there. So first of all, human influenza, first bacteria was done in 1995. Then the tuberculosis minimum genome was discovered. Then cerevisiae, Saccharomyces cerevisiae, and C. elegans, D. melanogaster, first eukaryotic. Then Homo sapien draft were given in 2002 or so. Then in 2003 we were finished with that. Then this is how the human genome data complete. Related to that, studies cumulative number of genomes were discovered, and this is for the eukaryota and this is for the archibacteria. So this is how bacteria has the highest genomes at the moment in our bioinformatical data. So 14th April 2003, the human genome was complete. You can see in the 9th May of 2000 Nature paper, chromosome 21 was discovered, and then chromosome number 22 in the Nature was discovered like that. So this is in 1990, and this is the Wellcome Trust first human genome project was done. So this was the books that have been established in the form of library. So all in 22, all the human genomes were sequenced like this. So we are done with this, with the human genomic project. How it was important, how many things were connected with that. So we have understood various graph and data related to that also. Now comes the new words like genomics. It refers to the analysis of complete DNA sequences. Then comes the functional genomics data, the study of obtaining an overall picture of genomic functional functions, including the expression of profiles at the mRNA level, protein level. Global approach are being used to understand the functions of genes and proteins. Proteomics is dealing with the refer to the study of proteins using technologies of large-scale protein separation and characterizations. Transcriptome dealing with the mRNA transcribed from cell genome, whereas the transcriptomics deals with the generation and studies of complete mRNA expression profiles. So structural genomics and proteomics, assignment of three-dimensional structure to the protein product of the genomes, and investigation of the biological implication of these assignments. If the structure assigned to a new protein is homologous to one already known, it provides an indication of its probable functions and evolutionary relationships. Then comes the genomics part, that the quantitative study of genes, regulatory and non-coding sequences. Transcriptomics we have discovered, proteomics, metabolomics, pharmacogenomics. It's deal with the quantitative study of how genetic affect host response to drugs. Physiomics, which deals with the physiological dynamics and functions of whole organisms. Transcriptomics, as we can see that in a probe dependent method such as microarray analysis, relative abundance of given label transcript is inferred with a fluorescent signal. So here we can see cell tissue or sample. We can have a cellular RNA out of it. Then we can do either microarray approach or RNA-seq approach. So in microarray approach, we label them with the cDNA and RNA, and then infer the presence of transcript later on. Then this one is the ATCGT RNA sequence approach, compare with the target genome and enumerate sequences. That we have done the DNA microarray approach. So we have a bacterial grown in culture, and we have the infected person, and we dye with them with the red dye and green dye, and then we check with the microarray with the red, yellow, and green which genes are expressing and which are not expressing as a white. In RNA-seq, we isolate all mRNA, convert to complementary DNA using reverse transcriptase, sequence the complementary DNA, map sequence of the genome. So then you do the fragmentation, reverse transcription, mapping, equintation is done. And in proteomics, in each part that we're studying here, so your bioinformatics is playing role actually. At the end, you have to bring out the bioinformatic part in your all these techniques. Here also, and you have a sample preparation, mass spectrometry, then and data analysis of your mass spectrometry. You have to do the analysis of your data. Proteome diversity, so from one gene sequence to one predicted amino sequence to many proteins. So here you can see that gene being predicted to the translated into gene products, co and post translation modifications of proteins, for example, N terminal or C terminal truncations, glycosylations, other modifications are done with the help of this technique. Systematic biology, here you can see that there's a planet, then there's ecosystem, then there's a community, then there's one organism, then there's systems, organ, cell, DNA. How all these parts are connected to each other. So how these protein interactions are being done. So to do so, human protein interaction could be done in order to check the interaction between different organisms. So top 10 challenges for bioinformatics are precise models where and when transcription will occur in genome, precise predictive models of alternative RNA splicings, precise model of signal transduction pathways, accurate ab initio protein structure predictions, determination of the protein DNA, protein RNA, protein recognition codes, rational designs of small molecular inhibitors of proteins, mechanistic understanding of protein evolution, mechanistic understanding of speciation, development of effective gene ontologies, and development of bioinformatics and bioinformatics curriculum. So that's it for today, students. Uh, So students, before you leave, can we have class tomorrow morning? At early morning. Do you have classes tomorrow also? Tomorrow is Saturday. Maybe your college is off. Can we do in the morning? So we can. Tomorrow I have to just teach you good scientific practices and applications of recombinant DNA technologies. Because in the evening I might be have some plans, so might be not available at the same time. So please confirm me some good time for you students. So you have classes, okay, okay. So we keep it at the same time. But what about we meet at 7 a.m. in the morning because we will be fresh. I might be. I will take 7 a.m. I'm asking so that no sir. So one is no. But these lectures will be recorded for you. So don't worry about it. You can watch them later also. So for you 8 a.m. the classes begin. 8 a.m. No, sir. So it seems difficult. So okay, we do the same time, then 5 p.m. then, and we meet us at the same time tomorrow. We do not change the time. But in future, I was thinking to change in the morning time so that no, it's not 7 a.m. It seems difficult for everyone, so I don't want to create a chaos for you. So we meet at 5 p.m. same time. So same time, same place. No confusion because you have few people here now, so I don't know how many people will join tomorrow. So I have to share in the group. So we keep it the same time, same place. So no confusion. At least everyone knows about that timing. So that will be not confusion. So today we learn about X-ray diffraction and a little bit about bioinformatics, how important bioinformatics is in various fields, from your proteomics till transcriptomics, promotobolome, gene array techniques, RNA-seq. So that's how we did today. Okay, on that note, thank you very much. Vielen Dank für euch für heute eine schöne Studentin ich wünsche alles eine viel gute Zeit und ja, auf Wiedersehen. Tschüss.